Good evening. I'm Jerry Rubin. I'm the director of the Janelia Farm Research Campus, and on behalf of Howard Hughes Medical Institute, I'd like to welcome you to tonight's lecture in our Dialogues of Discovery series. Tonight, we're very fortunate to have John Donoghue here from Brown University. Yes, very complicated. John is a very hard name. I have a lot of problem pronouncing names. Usually John, I'm okay with John, but you know, even John is a professor of neuroscience and engineering, and you're going to hear tonight how he combines those two disciplines in a very fascinating way. And I'm not going to give you his whole biography because you all got a copy of this little pamphlet when you walked in that tells you where he got his degrees and about his research program and background. So I'm just going to invite John to come up here and talk about into the heart of the mind, neurotechnology to restore lost function.
Thank you, Jerry. Oh, I have a microphone too. Thank you, Jerry. It's really a great pleasure to be here, and it's an honor to speak in this series. It's really a wonderful thing to reach out to the public, and I'll do my very best to make as much accessible as possible to a very diverse audience. So what I'd like to talk about tonight is really the emerging technology, neurotechnology revolution, where there are devices that are being developed and really appearing at a very rapid rate that allow us to interact with the nervous system, to perturb its function, to change its function, and also to read out its activity. And what's really amazing, and I'll spend most of my time on, is that there are devices coming that can restore lost function, that allow us to actually make replacement parts for the brain and fix things that we never thought we could fix. And along the way, we're also having a new peek inside the human brain and beginning to understand with these new neurotechnology tools, which are really clinical devices, but they're also research devices that are helping us understand how the human brain works. And the model of this is something that I think is well established and you may not recognize. So how many people here know what that is? Good, it's not easily recognizable. That's one of the first cardiac pacemakers. And it's typical of how technology, medical technology devices that are inside the body are developed. When this was first developed, this gentleman had a cardiac arrhythmia, and they wanted to repair it by electrically shocking the heart to get it to have a normal rhythm. And because they didn't know how much to shock it or how often to shock it, they had a big stimulator on the outside of the body and a big recording device that allowed them to see the heart waveforms. And he basically had a couple of wires going through his chest that had a bandage over them that went inside his body. So this is a classic engineering device for early stage when you don't know what's going on. You keep everything on the outside. You put as little as possible on the inside. But of course, as I think all of you know, that the cardiac pacemaker of today, in which there are hundreds of thousands implanted almost without thought, when you need in a cardiac arrhythmia, this is a life-saving device and a life-extending device. And it is a sophisticated technology that has, over the course of several decades, now almost 60 years, become a device that's a regular everyday device. So neurotechnology is more like this. And you'll see some examples that are a little bit mature, but a lot of it, especially, I'll talk about my own research, that is about technology that seems to be back in this early stage. And where this is going is really exciting. So you can define neurotechnology in different ways. There are kinds developing that are labeled by where they are. So there's a bunch of them that are outside the head, and there are a bunch that can go inside your head. So outside the head ones tend to be big. Some of them are familiar, like an MRI machine, which allows you to peer inside and look at the structure and even function of your brain. You can stimulate the brain with a magnetic coil on the outside. You can read out brain signals with these kinds of devices on the outside. These devices tend to be gross, crude, and generalized either measurement devices or generalized perturbation devices for the brain. And there's even technology that I'll just mention maybe again at the end. There's a consumer product of sort of trying to read out activity, but it really doesn't. So these devices are on the outside of your head, and as I said, they're really not, they have some diagnostic abilities. They have some interventional abilities to help people with disorders. But really, the big revolution is happening with devices that are placed inside the head. And this is a lady that has a device implanted in her brain. We'll talk more about devices like this in a second. She has Parkinson's disease, but you wouldn't know it. So that's one advantage of these kinds of devices that are inside the body, is you can't see them. And they're being placed acutely, which means just in a surgical procedure to figure out what's going on in the brain to guide a surgeon. But also more and more devices are being planted chronically or permanently implanted and left in the brain. And you'll see all these things. I won't tell you what they are now, but they're to be able to perturb the brain or to read it out. So another way to think about the kinds of devices there are, and I'll only talk about these, what we call intracranial, inside the head devices, are those that stimulate or write into the brain. They can electrically stimulate the brain and influence its activity. And these are being used to restore lost sensory function. I'll give you an example. And for therapy called neuromodulation, and you'll get to see what that is in a second. And the other, and in fact, much more early stage technology is to read out what's going on inside the head. And that's being used to detect or predict abnormal activity, as in epilepsy, but also to restore movement, which is what I work on, and I'll spend the good part of the talk telling you about how you can read out the brain to help restore movement for people with paralysis. But let me give you some examples of writing into the brain and how that's being used. So probably the most successful device like this is the Cochlea Implant. So the cochlea is this little curled up snail-like device that's deep inside your skull. It's got a big nerve that connects into your brain. And in many people with hearing loss, in fact, most people with hearing loss, they lose the little hairs that respond to sound that allow you to hear. It transduces the sound into electrical impulses that go into your brain. And the Cochlea Implant is a device in which a surgeon places a coil with 22 electrical stimulating sites inside this little snail cochlea. And due to an amplifier on the outside that broadcasts to a sensor on the inside, these 22 channels can take sound and transduce it into electrical tickling of the cochlea that you can interpret as a sound. And there are hundreds of thousands of people, including children, who have begun to hear, who can hear as a consequence of these devices. Now, I can play you a couple of sounds of what it's believed, because we don't have direct reports, but it's believed what it sounds like when you first turn it on, and then later after people hear for a while. And this is a case where early on people get an unusual pattern of activity. We're getting 22 channels of sound input when we have 40,000 input channels. But they hear something, and yet the brain is adaptive enough that after a while it begins to understand that these complicated and not so clear-cut patterns of activity really mean something. So here's what it sounds like when it's first turned on. And this is a synthesis that people believe is what it sounds like. Doesn't sound like much. But that same sound, and it is actually a degraded version of this next audio clip.
She's drinking from her own cup.
So not completely normal. It's kind of tinny sounding, but nevertheless, for people who can't hear, this is an amazing advance. So this is one kind of interface with the nervous system, and as I told you, there's about 200,000 people with devices like this, and it's a commercial device. Another stimulating device that's around is a retinal implant to restore vision for people who have lost vision. The same thing is true in many forms of blindness. People have lost vision because the photoreceptors, little receptors inside your eye that pick up light, degenerate. And the idea behind this device is to have a camera that picks up a visual stimulus. It gets converted into a pattern of impulses, and those pattern of impulses are converted to electrical impulses on a little patch of electrodes. So this patch has about, depends on the device, but about 64 sites that are little tickling sites, little electrical stimulation sites that stimulate the pathways that go into the brain that remain. Remember, the receptors are gone, but the pathways are still there. So it tickles them, and as a consequence, people can see. But of course, again, like the cochlear implant, it isn't the kind of vision that you or I have. So it's something like this. So first, here's the actual device. This one has been approved for human use. Here's the actual device implanted in an eye, seen through a microscope. There, each one of those little metal reflections are the stimulating sites. And so, as I said, the vision isn't normal. It's something like this, but you can see what this is. What is it? A hand. So even a very degraded version of an image gives you something. And here's a lady using this device, and you'll see a couple of people who are using the device. And again, you'll see that it isn't normal vision. This is where we are in the field of being able to restore some sight to people.
This
this one up here,
Uh-huh.
is that the circle?
Why don't you put your hand up there and confirm that? Good. Put your hand up there. Touch it now. Remember our...
It's like I'm trying to touch it. It's going to bite.
Remember our hand coordination practice. Now, good. Take it.
So she can see something. Again, here's a man trying to see a spoon on a table. Notice he's going back and forth to get that kind of image going across his retina, and yet he can see. So this is, again, an early stage device. This is not what we would call everyday vision, but this is for a person who can't see at all. This is a remarkable advance. Now, another remarkable stimulating technology is involved in what I told you is called neuromodulation, which is rebalancing circuits. So this lady has a disease called Parkinson's disease. In Parkinson's disease, you have a loss of a chemical called dopamine in the brain, and it gives rise to symptoms in which you're rigid, you shuffle when you walk, and you have shaking. You shake repeatedly. It's called the shaking palsy. And the reason you have this is you have circuits that go from the deep parts of the brain to the superficial parts of the brain, and they're bathed in this neuromodulator dopamine. This chemical dopamine is bathing this circuit all the time, and the circuit needs it to work normally. And when you lose dopamine, the circuit goes out of balance, and you get these symptoms. Exactly what goes on, we're not positive. However, it was discovered in the 1980s that if you take a stimulating electrode, so this is an X-ray of a person's head, here's this sort of ballpoint pen refill diameter kind of flexible plastic tubing with stimulating sites that are placed in the brain by a neurosurgeon, trying to hit a target called the subthalamic nucleus. It looks almost exactly like a Tic Tac candy. It's about the same size. It's deep in your head, one on either side. And when you put these electrode leads into the subthalamic nucleus on both sides and you turn it on, you rebalance the circuit. And here's this lady with the stimulator. So so this is a person who has a brain implant, bilateral, has had surgery to place it in there. How many of these do you think have been put in? I just always like to get a guess from the audience. 10,000. How about, well, actually, I think I have 80 on here. No, 100,000. It's more than 100,000 worldwide. So the surprising thing is that this is a regular procedure now. It's done very frequently in most academic major medical centers, and it has very dramatic effects. So it's rebalancing circuits. And I have to say that some of the work that's going on here at Howard Hughes is very important in understanding how circuits work and how dopamine and other chemicals can influence how circuits work, because we just don't understand exactly what it is that causes this balance or rebalancing of circuits, but this is something that needs more scientific inquiry. Another very dramatic example of using the same kind of electrodes, deep brain stimulating electrodes, in a different target is in what's called DYT1 dystonia. It's a genetic type of dystonia. Dystonia is something where the muscles are tense all the time. And this young girl who you'll see, I think I can play the video, has dystonia, and you can see her neck. She can't move her neck out of that posture. See how she's twisted around in her wheelchair? She's really stuck like that, and it's a genetic disorder of muscle tone. Now, with deep brain stimulation in globus pallidus, you can see here she is after the stimulator is placed, and she's walking, and it really has a remarkable effect on liberating you from this horrible imprisonment in your body with this increase in tone. So these are two examples of where this kind of stimulating technology to modulate the brain back into some kind of normal function has had a big impact. So I've talked about stimulation. Now the other kind of neurotechnology that's developing is for sensing or reading out signals. And what I'm going to talk about is my own work to restore movement. So this is much earlier stage work and is something that is being used to reconnect the brain to the outside world for people with paralysis. So here's a young man playing a video game. He's supposed to hit these targets and avoid these goblins. These are treasure chests. And he's playing the video game, moving a cursor. And you say, well, he's pretty good at it for a guy. He's in his 20s. You can't see him very well. But this is Matt Nagel. Matt Nagel, in his early 20s, was stabbed in the neck, and it completely severed his spinal cord, so his brain is disconnected from his body completely. He's totally paralyzed. And we have put a tiny chip in his arm area of motor cortex. By the end of the lecture, you'll know what that means. We've put it in this arm region, and we're taking the signals out from his thoughts about moving, playing it through a computer that's decoding it and sticking it into this computer. And so when he's thinking about moving his arm, he's actually controlling that cursor. So one of the reasons why he's not great at it is because we're using a tiny sample of his brain's own signals to control this cursor, and we're guessing at how to make sense out of the brain signals. I'm going to tell you about how we do that. So these kinds of devices are called brain-computer interfaces, or a neuromotor prosthesis, something I hate because it's harder to say than John, and much harder, or brain-machine interface, and machines kind of conjure up all kinds of strange images. But so we've called this technology BrainGate, and it's a kind of neural interface system. So first thing I want to remind you of is what paralysis is. And paralysis isn't a disease. It's a consequence of something else going on. There's a whole list of disorders that can give rise to paralysis. And what happens in many cases of paralysis is basically you have an operating brain that is disconnected from the body. Now, in some cases, it's disconnected, say, by a stroke, which is the blood supply either has tissue that dies or it interferes with the tissue. And it cuts a pathway that goes from your brain to your spinal cord of about 1 million thread-sized wires, really tiny wires that connect the brain to the spinal cord. So the commands, I want to move, can't get there. And the most devastating kind of this stroke, and you'll see Kathy Hutchinson later, is up high in the brain stem above the spinal cord, and you can't move your body at all, and you can't even speak as a consequence. Another kind of disorder is that causes paralysis is something like ALS, which is a degenerative disorder, Lou Gehrig's disease, in which the connections between the spinal cord and the muscles degenerate. And in that disorder, the messages can get to the spinal cord, but then they can't get to the muscles, and so you can think, but again, you can't move. And finally, we can even think of limb loss as a kind of paralysis because you have a brain that works, but you don't have a body to move. There's nothing there. So a BrainGate or a brain-computer interface could help all of these people if you could reconnect the brain to the outside world. So I think many of us don't appreciate the scale of paralysis in the world. Christopher Reeve Foundation estimates there's about 5.6 million people living with some form of paralysis. It could be something from total loss of independence, which we call tetraplegia, 4 limbs lost function, or with anarthria, which means they can't speak either. In the worst case, it's called locked in, which means the person can only move their eyes vertically. They can do nothing else. It's the only way to communicate. And then there are other people that have much more limited loss. This is a gentleman who had a stroke. Is actually a person who grew up with cerebral palsy who lost the pathway from one side of the brain to the body. So he has this paralysis on one side of his body, has this characteristic flexed arm and leg. And these individuals like this have reduced independence. They have what's called paraplegia, which means 2 of the same limb don't move like their 2 legs, or hemiplegia, which means one side of their body doesn't work. And as I said, some people have limb loss. And there's really quite a few people who are in this kind of a population. So there's about 12,000 people a year have spinal cord injury of various types. About 750,000 have strokes. And of those, about 130,000 have the inability to move their arm in a functional way, one arm in a functional way. So the point here is that if you could reconnect the brain to the outside world or reconnect the brain to the body, it would really be something very beneficial to a large number of people. So I want to point out that movement, if you want to restore movement, it's really a daunting task. This is just a video that my kids gave me of that I think all the high school students we talked to today are all YouTube fanatics, and they've seen, I'm sure they know this one. I had never seen it before. But this young kid is really pretty remarkable. But it shows you how unbelievable our motor system is. He's integrating information and moving and doing all kinds of tricks and things that I will never be able to do, I'm certain. So it's a hard problem. So here's Asimo, the robot, able to walk around and do things. But it's really hard. Watch Asimo trying to just go up a flight of stairs. And, you know, if you don't have everything just right, there's a real problem. And, of course, we grow up with that problem. You know, that we spend a large percentage of our early life trying to figure out how to see something and then get it, you know, and here's a young guy struggling to actually get that arm to go where he wants it. So we're faced with a big, daunting challenge. So what I want to do then is sort of give you a sense of how could we think about connecting the brain to the outside world to create action directly, or to connect the brain even ultimately back up to the muscles, you know, and be able to redo this with physical components. So this presents a whole bunch of problems, and these are questions I want to answer for you. What area of the brain would you get signals like this from? what signals would you even want? You know, what could we get a signal out? And if we did get a signal, do we have something that can pick it up, a sensor that can pick it up? And if we get these signals out, how could we make sense out of it, translate it from brain language to control language, say for a computer or robot or something? We call that the decoding problem, to decode the brains or translate the brain signal. And then what would you do with it if you had it? If you had these command signals, would they be good enough to do anything like control a complicated arm, or maybe something simple like Matt controlling a cursor on a screen? So let's answer these questions. So the first is, where would you go? Well, we've known since about the 1870s that there's a part of your brain that controls movement. It's called the motor cortex, and it's a strip on the outside of your brain going from about the top of your head down to your cheekbone, about an inch wide. It's broken up roughly into thirds. The top third controls your leg, the middle third controls your arm, and the most lateral part near your ear controls your face. And we've known for over 100 years, for example, if you have a stroke and it hits that area, you can't, for example, move the arm if that arm area is damaged. So we've known that we know this topography, we know this region to go to. In addition, we know something about the signals that come out. The signals that neurons produce are called action potentials. They're very brief electrical impulses, these vertical lines that all us neurophysiologists watch all the time. And what they are is the electrical impulses that are coming out of a neuron that are actually traveling around the nervous system, and it's the communication language of neurons. So you can think of neurons like little radio broadcast towers. In fact, some of them even look like radio broadcast towers. And they put out these electrical impulses, and the problem is that they only travel a very short distance. In fact, if you don't have an electrode that's very fine and nuzzled up close to the cell, you can't even pick them up. That is, so they're not outside your head. They're just very tiny signals that are very difficult to get. And we can think of them kind of like a series of ones and zeros that come out as a pattern. So each of these impulses is like a pattern coming out, and I'll explain how we make sense of those patterns. So the problem is even worse in that one neuron is not all that important, fortunately, because probably during this lecture I'll lose a half a dozen neurons or more. As you age, they begin to die, but your brain is robust and can deal with that. So there are, even within the size of a pea, there are thousands of neurons. You have about 1 trillion of them in your brain. But they work together. They don't work independently. So not only do we have the problem of recording from neurons and picking them up, but we have to pick up many, many neurons. And the neurons put out patterns of activity. And these patterns are the code, the real code, these patterns that are coming out. And you can think of these patterns as something like QR codes. So I think everybody here, I hope, has heard of a QR code. It means quick read. They're the things that you see on the side of your soup cans and cereal boxes, and you can have a smartphone decoder that can decode these. And I think of this as a patch of brain activity. And each one of these black or white squares are like those action potentials coming out of a single neuron. So each little square is like a neuron sending out its impulses. And unlike a QR code that just sits there, your brain, each little patch of your brain is flickering about 100 times a second with a pattern, a QR code. So the job of decoding is trying to take that thing and make sense out of it. So in order to get that kind of a pattern out, we had to invent a microelectrode array. That is something that you could put in the brain and pick up some percentage of these neurons that are doing something with movement. So we knew where to go. We knew it would go in the motor cortex. And we came up with this device that was originally developed by Dick Norman at the University of Utah, and we worked together to develop it so it was human compatible. So it looks a little big there. It's not that big. It's about the size of an M&M. It's 4 by 4 millimeters. And that device is implanted into the arm area of motor cortex. These are my fancy graphics for PowerPoint keynote. So it's implanted into the brain. And so here's the cortex. It's about as thick as an orange peel. The cortex is the outside of the brain. Here's the region, the gyrus, the bump in the brain where the motor cortex is. And here's the electrode array, and you'll actually get to see a surgical procedure where we put it in a couple of minutes. It's put in now in our current version. There's a cable that comes out carrying the 100 signals from this microelectrode array, carrying it out to a plug on the head, and these signals come to the outside. As I said, we're in the early days. We want to take everything outside because we don't exactly know what to do with it. Okay, when we get these outside, how can we make any sense of these impulses that are coming out? So let me start with just giving you an example of one neuron and the model of what that one neuron might be coding. So imagine we have an electrode, just one in my brain, and we're recording from one neuron. And I reach to the left, and when I reach to the left, the neuron fires one impulse, one spike, we call it. And then I reach to the right, and it fires 5 impulses. Okay? So now we could say, oh, we have a code. 1 means left, 5 means right. So later on, we see the following. Okay, you're recording from me. I'm behind the stage. I move, and we get 1 impulse. Which way did I reach? Left, right? So you're a decoder. That's all we do is we say, okay, in this little time interval, we saw 1 spike, so it's left. Here's another one. We saw 5 spikes, so it means reach right. So that's basically the idea. Of course, it's a lot more complicated because we have a lot more signals, and the signals are actually noisy. We can't rely on 1 always meaning left and 5 always meaning right. Neurons jitter up and down and do all kinds of much more complicated things. But that's basically what we do. If we see a pattern by recording all of these, and this electrode has got 96 channels, we pick up 96 channels, we get a pattern of activity. And one pattern we say that means right, and the other pattern means left because I've moved to the right or I've moved to the left, and we can look at the patterns that are generated. And then in the future, whenever we see this pattern, we tell the computer that is reading this out, whenever you see that pattern, move the arm, the cursor to the right, or move the cursor. If you see this pattern, move it to the left. You got it? You're all decoders now? So you can all be neurophysiologists and put me out of business, see? Okay, so in the early 2000s, we put all of the system together, and I'm neglecting a large number of years of research, what we call preclinical research in animals that led to safety testing and efficacy testing that said that we could actually do this in humans, and then a process where we went to the FDA, who's here with us tonight. Where did you guys go? I don't know where they went, but they were here, and we worked with the FDA to come up with do what's called a pilot clinical trial, and this pilot clinical trial, we were then given permission to test the device in a small number of people to understand its potential, certainly its safety, and the potential for this to become a useful brain-computer interface. So here is the array of electrodes that we developed. It's implanted into the arm area of motor cortex. The plug on the head provides signals that come out. It goes through a bunch of signal amplifiers and a cart full of electronics. It goes into a computer so the person thinks and moves a cursor because we decoded it properly. So all of that should make sense, from array to motor cortex to pattern to cart of electronics and cursor, right? Everybody following me here? A lot of technical stuff, but you're actually pretty good neuroscientists if you're going this far. This is sort of at the edge of where we are. Notice the cart of electronics looks a lot like the old cardiac pacemaker idea. So that's sort of where we are again, because we don't know exactly what we're doing. So far in our clinical trial, we've implanted 9 people. 2 of them had spinal cord injury, 2 with brainstem stroke, and 5 of them had ALS, as I told you, Lou Gehrig's disease. I'll mostly talk about Matt here, who had a spinal cord injury, and Kathy Hutchinson, who had a brainstem stroke. So Matt could speak, Kathy could not. And we have about 6,000 days total experience across all these people, and it's been very encouraging, but it's an investigational study, so that means that we are examining this, and this is the data that we're putting together that will report on the safety and performance of these devices. So, okay, if you don't like surgery, you should close your eyes. Here's the actual implantation, and you know, to a neurosurgeon, this is a very simple procedure. First of all, in an MRI, we find this little bump on the brain called the knob, which conveniently turns out to be, in a picture of our living brain, turns out to be the place where your arm is controlled, so we can actually see it in just a static MRI picture. The surgeon makes a hole in the skull about the size of a 50-cent piece. There's a membrane here that's open called the dura, and the array is placed. And actually, it's tapped in because it's a little bit hard to implant it just without pushing it in. So you'll see the surgery. And the tap is just about there. It was. That was the tap. So it's not a big event for the surgeon to put it in. It's now on the brain. All the membranes are closed. All the bone is put back. The skin is closed, and all that sticks up is the little plug. And the person, we can ask the person, does it feel like anything? And they don't really feel anything at all there. So the first question that came about after we implanted these arrays is, in a human who hasn't moved in many years, and all of these people, it was at least 2 years, and in many cases, multiple years, does the brain actually still work? This is the part of the brain that controls the arm. They haven't controlled their arm in years. Does it still work? So I'm going to play you a video in which you're going to hear a technician saying to Kathy, imagine opening your hand, imagine relax, and then imagine closing your hand immediately after we turned on the system. And what you're going to hear, and now you can hear the action potentials because I'm going to play them through the audio monitor. You can hear the spiking. And you can now think, how does the spiking relate to what she's doing? And you can hear for yourself that it's really quite robust.
Relax. Now imagine you're opening your hand. Relax. Close your hand. Relax. Open your hand.
So I think you also understand why neurophysiologists always talk about hearing neurons. You can't really hear them, but when you play them through an audio monitor, they make that sound. And you can hear that there's a big difference in the spiking. So again, that's what the computer is doing. It's counting up the spikes. And even with that simple one cell, if you had a lot of spiking, you could open a hand. And if you said low spiking, you could close the hand. So in fact, that's what we did. Here's just another video that I like to show of Kathy. We just had her play a game where every time this turned green, she was supposed to, her spiking made the knee. Now, what's impressive about this is that immediately upon turning on the system, the brain was working and producing as if it was actually controlling a hand, even though nothing was happening. She couldn't move at all. So this meant that the signals, not only are they there, but they're preserved in a way that they look like they're controlling an arm. So that means we have a source of command signals to be able to control devices. So as a consequence, you've seen already Matt playing the video game here. We could take those spikes out, and I won't go through all the details, but we just had Matt and Kathy and the others control a cursor on a screen. And but we did that by, again, building a decoder that took the left and right and then up and down of all the coding and just use that to drive the cursor around. And here you can actually see Matt drawing a circle, and he'll comment on what it was like. So Matt's drawing a circle. First neurally drawn circle.
Oh man, I can't put it into words. It just, I used my brain. I just thought it. I said cursor go up to the top right, and it did. And now I get control of it all over the screen. It's wild.
So he actually had no real insight into how he was doing it. He just controlled it. So I'll play a video. If anyone objects to an expletive, I will turn the sound off. Does anyone object? If anyone objects, I will turn it off. Okay. So because this device, he's going to control this robotic hand. Again, it doesn't really do anything. He had a direct brain control of the robotic hand, but it shows you how important or how powerful this was for Matt to be able to control something. In this case, it was, I think, a couple of years after he had controlled anything. But just listen at the very beginning of how he reacts.
Close. Holy shit. Open.
So he was really taken. And then Kathy, we had her set up to drive a wheelchair, but actually I think the control is not good enough that you would have her driving this wheelchair around on her own. But she was using this interface to control this wheelchair. She actually ran over my toe and hit the bookcase. So she's kind of a dangerous driver in this. But it's just to show you that we can. These are only demonstrations of what you might be able to do with this kind of technology. I think so really what you'd like to know is what can you do that's useful? So that's what we've been doing over the past years. One of them is if you could control a cursor, you can type on a computer screen, or you can do anything that a computer can do just by moving a cursor. And here, Kathy is doing point-and-click typing. And because it's sort of like typing with one finger, it's a little bit difficult to type on a keyboard that's a virtual keyboard on the screen going back and forth typing letters. And those of you who are old enough to know about original typewriters, the keys were set up in a way to alternate hands, which means you really had to move a lot if you're just using one finger. So we made it what's called a radial keyboard for her. And here she is typing, and it does like your smartphone does. It does some word prediction. And so we're developing, and I won't talk much about this, but we're developing these brain interfaces for communication. And that's especially important for people like Kathy who can't speak. And it's valuable for her to be able to say to her family, I'm in pain, I'm uncomfortable, I need some medication, or I'm hungry. So we're working on that kind of a device. Now, we can also build devices that are kind of an assistive technology, like a robot arm. Here's a cartoon of what we would like to do. Here's a person, and now in this case, the BrainGate sensor is just this glowing dot. It's radio magically transmitting this to the robot arm. And then he can have a drink of water as a consequence of being able to control this arm. So one of the questions is, can you, in fact, are there such robot arms available? And the answer is there's really quite a few very sophisticated robot arms. Here's one that's developed by the German Space Agency, DLR. It's their collaborator, Jorn Vogel. And this is one that's actually very safe to interact with. You can bump into it and it stops softly. And you can show you how compliant it is. So there is such a device. So you could develop a robotic assistant. You could replace lost functions. So in a person who has lost a limb, you could build a robotic device that could be controlled by the brain. Here is a gentleman controlling a prosthetic limb, the latest versions of a prosthetic limb that's being developed by DECA and DARPA and the Veterans Administration for people who have lost their forelimb. But this forelimb is being controlled not by the brain, but by accelerometers on his shoes. So he wiggles his feet, and that's what controls it. He actually does a pretty good job. But imagine now he could control it with his brain. It'd be like just being, having a real arm that you control directly yourself. And then the third kind of dreamlike scenario would be for restoring arm function is to have something like this, a BrainGate sensor that decodes brain signals and feeds it into an implanted stimulating system that's in the arm. So imagine a stimulator that receives these signals and converts it into patterns of electrical activation through wires, and those patterns drive the arm in a natural-looking way. So, and the person then could use their own arm. So this isn't quite so fanciful as you might think. The technology, which is called functional electrical stimulation that stimulates arm muscles, actually exists. There are about 600 people with various versions of this. And the FES system, here's a gentleman who has that device implanted. He's using a switch on his shoulder that he can wiggle his shoulder and control it. So you can see, again, it's not elegant, it's not perfect, but he's got a little remote control over here that he can pick up using these controls. Now imagine he had brain control of that arm. He could now have his physical nervous system back again. So that's the kind of thing, and this is our ultimate aspiration, is to move towards being able to give people back control of their own arm. So where are we? So let me show you where we are in controlling a robotic arm. So we're looking at how can we take brain signals and control something as complicated as an arm, but we're using a robot arm mounted on a tabletop, and here's that DLR arm, and here's Kathy. And again, now you can see what the real scene looks like today of Kathy in her chair. Here's the plug on the head with the little amplifier cables coming out to the electronics that decode things, feeds a signal over to the robot arm, and we asked Kathy to control it. So we built a decoder that mapped, when she imagined moving the robot in different dimensions, back and forth, up and down, we built a decoder, like a QR code for it, for each of those actions. And we also built one for when she imagined opening and closing her hand. And we asked her to do a number of things that I'll show you. First, we started simple with two-dimensional movements and then did more complicated movements. So here, we asked Kathy to pick up a wine glass and then put it down on this little tablecloth that had circles on it, says one of the targets. And so here she is picking up the glass. She only has two-dimensional control here. When she closes her hand on the glass, it actually lifts it up, or when she tries that again, it lowers it down. So she puts the glass down. And she communicated with us later that after she did this, and you can see Patrick, our collaborator, coming over to get the glass. And she says, nope, I didn't get it quite in the right place. So she went back over and moved it over in the right position. So she had two-dimensional control, but the real ambitious thing was, our arms move in much more complex ways. So here, and in fact, this is a slightly different robotic arm. We asked Kathy, we built a decoder for 3 dimensions plus grasp, but we asked her, we had these balls on elevated sticks that put the balls up in the air. It's actually a very difficult task to do. We've tried to use a mouse to control this and move the arm to grab these balls. But we said, Kathy, try to squeeze these balls and that are elevated as they're, and you can see she's moving around. And it's quite hard because the wrist here does not move yet. And so she has to orient the hand in a way that it'll actually engage it perfectly. But she can reach in 3 dimensions using just this little patch of brain, this little 96-channel electrode array. And no, so I don't want to deceive you. She's not moving like you or me. She's moving much slower and with much less precision. So this is a complicated graph. In fact, I'll just summarize it by saying we asked her to touch the ball or grasp it. So she and another patient they could at least touch it about half the time. And one of our participants was getting up close to 80% of the time. But then actually successfully grasping was a significantly smaller percent, about half in the best cases, about half the time. So we're not perfect in getting really good control. And it takes a long time. It took, between 8 and 18 seconds. It takes a long time compared to what we can do, reach out and take a glass of water and drink it. This is pretty slow. Nevertheless, if you can't do anything with your arms, you're completely reliant on other people, this is still a major advance. So Kathy, we asked her what she would like to do, and she said, I just want to serve myself my morning coffee. So this is how she served her coffee, this bottle with a straw. She is a little bit weak in the way she can drink, so she has to have a straw. So we said, okay, Kathy, for the first time in now 14 years, your job is to give yourself your morning coffee. So here she's controlling the robot arm and bringing the coffee over to her mouth and taking a drink. And so again, you can see that it's slower. It's certainly not as smooth as what we would do, but nevertheless, she's doing this on her own under brain control. We actually had to tell her, okay, Kathy, you had enough. Put it back. And then we also found out later that in some of the trials, she had Kahlua in the coffee as well. The afternoon coffee came with Kahlua. And again, small steps, but certainly a big step for a person she had to be careful there not to knock the bottle over. And this made her extremely happy. So now, I've shown you some what I think are pretty amazing things, but there's really a long way to go here. And so one of the things we have to do is sort of get rid of all of this. We have to get rid of this big cart of computers, the technician that's in the room every time we run a session, and we have to get rid of all this wired up stuff on their head so they don't have a plug on their head and are tethered so they can't move around freely. So again, this is our vision of what we'd like to do, is implant the array completely inside with a wireless transmitter that would transmit the signals from under the skin to a device worn on the body that would then control, say, an FES system. So we are actually making progress in that direction. And thanks to my colleague Arto Nurmiko and his student David Borton, who's now a faculty member at Brown, they developed this implantable device for certain generations. You'll know what a Zippo lighter is. You'll know the size of it, or a Matchbox for others. Both of those are, I think, going out of existence. But this device is a 100-channel wireless amplifier system that is implanted so that the array goes in the brain, the wireless transmitter goes just under the skin, and it transmits all of those signals outside the body. And we have this, actually, this is a real one that functions. It is now being tested in animals, but is not yet ready for human application, but that's where we're headed, so that it would, in fact, untether people and allow them to use this device all the time. So you could ask, what's left to do? Well, one of the things we want to make sure this is safe. That's one of the things we're testing in what we call a pilot trial, a reliable. That means it has to work all the time. And we have seen that there are certain improvements that need to be made, that it's always available. Without a wireless system and a plug on the head, it's not always available. Portable. Cabling is not something that's amenable to everyday life, to be cabled to a cart of computers. To be affordable means that we have to find ways of making this so it isn't so expensive that no one can afford it. And of course, it's important for the users that it's useful in everyday life. So we have a lot of things to do. We have to, in our sensors, we want to make sure that they are biocompatible. That means the brain accepts them for long periods of time. They don't break down. They don't move around. For decoding, you know, I told you we're slow and inaccurate. We want to make them much better. And that's a neuroscience problem. That's what we need to learn from understanding how brain circuits work, is if we understood exactly how those spikes, that complicated pattern of spikes, was producing arm movement, we could in fact replicate it, we believe. So we need more neuroscience to understand how to make it work. We need these smaller processors that I showed you. And of course, we need to make sure that there's this sort of cheap portable robots available to people so they can control them, or FES systems, which are currently still very expensive devices. So one metric to say, where are we in the world, is to compare us to science fiction. So I like taking this. So here's Kathy today. And here's Star Trek. I hope most of you remember Star Trek. And this is Captain Pike, presumably 1,000 years in the future, but I heard it's not exactly 1,000. And Captain Pike is in a brain-computer interface device, and I'll just let you hear the interaction when he uses this to communicate with Captain Kirk and Mr. Spock.
You remember these gentlemen. They wanted to visit you. 2 flashes mean no. I thought you might make an exception for them.
So this is the ultimate horrible decoder. After 1,000 years, 2 beeps mean so this is really, really disappointing, you know. So by those standards, we're way ahead of them. We're 1,000 years in the future. Now, but then along comes Star Wars and ruins it, because we don't really know when Star Wars takes place. But here's Luke, as you remember, had his hand chopped off and with a lightsaber. And here he is in a non-sterile surgical room having this artificial hand put back on. And not only, we don't have any sensation in any of the devices we have, not only can he move in a fine, dexterous way with an instantly attached device done by a robot, but he can also feel. So that would be wonderful to be able to do, but we're nowhere near that kind of elegance yet. So our goal in the next years coming is to simply have someone who can do something better than this, we hope, but be able to power their own arm with an FES device. So we have a ways to go yet. Now, so just to summarize all of that's going on in sort of this neurotechnology revolution, I told you a lot about stimulation and all the different kinds of control we can get with neuromodulation, which is being applied not only to Parkinson's disease and dystonia, but also to psychiatric disorders where maybe we can stimulate the brain, and this is being tried in depression, where stimulation can help overcome depression or a number of other disorders. Hearing, I told you about vision. So there's a number of other applications of stimulation, and so this is really going ahead very quickly. I talked about BrainGate and being able to restore movement from paralysis. It goes has a ways to go. There are other devices. One of them that's coming along, there's already a simple device to treat epilepsy, and by treatment what I mean is it has a sensor, not quite as complicated as the one I showed you for BrainGate, but it picks up activity in the brain when a seizure is occurring, and it uses electrical stimulation like the DBS electrode I showed you and tries to stimulate the brain to stop the seizure. That has been FDA approved now to use in certain kinds of, to use in epilepsy. Now, so imagine in the future a better device that's very sensitive to spiking patterns that can predict, like the thunderstorm you heard coming in, you know, it can predict that a thunderstorm is coming, the thunderstorm of electrical activity in the brain, which is epilepsy. It could predict that coming, and it could now deliver a drug in a focal area, or it could tickle the brain back into normal activity in the area where the epilepsy is happening, all unknown to the person who has this device. A bit fanciful, but that work is already ongoing to see if this is feasible, and it actually looks fairly encouraging. So these kinds of devices are also coming and promising, and so I think there's really an enormous amount of exciting future for neurotechnology. Also, I mentioned human neuroscience. We're learning a lot about the brain. I'll give you just one example, and that was the example of plasticity. We had anticipated that in the motor cortex of people who hadn't been able to control their arm for a long time, that the area might be deranged, it might not work properly, but in fact it seems to be working just fine, and we're puzzled. Where's plasticity? So we have a science finding that needs to be investigated. Why is this human brain not reorganizing when it's not being used? We have many bits of data from many sources that if you don't use the brain or it's disconnected, it reorganizes. It doesn't appear to have done that here. So there are many interesting neuroscience questions that could be addressed. I also want to leave you by warning, watch out, that all of these devices, especially the ones on the outside of the head, are so easy to make or use that they're really popular. So see, here's brain stimulators that actually work. And so you can see the nation's top doctors have clinically proven the Fisher-Waller stimulator, which will cure, I think, insomnia, anxiety, depression, and pain. And I'm just warning you that you're going to see all kinds of devices like this and all kinds of claims. And if you're good budding young scientists like our students that are here tonight, look for the data. That's the important thing. So that's a warning. So where we're going to go, we don't know. It's like the telephone was in 1876 when Bell invented the telephone. Western Union said, oh, it's a fluke. Nothing will ever happen. Now imagine what Bell would have thought if he knew where the telephone was going to go. And after 130 years, we have smartphones that tell us everything. So I think the future for neurotechnology is a little bit difficult to chart out. Bell certainly wouldn't know what kind of apps would be available for iPhones. So we don't know where it's going, but it's a very exciting future. And finally, I just want to close by thanking the team of people that are involved in this research. It's not me. I'm one person that's part of a large team of extraordinary people. I mentioned Arto is doing the engineering. Michael Black, Wilson, Trucolo, and Michael Black are doing a lot of the computational work. Carlos has done a lot of background computer work and also work with the monkeys that led up to this work. And my colleague Lee Hochberg, who's a clinician scientist who is leading the clinical trial and is a brilliant young neurologist and neuroscientist. And we have now expanded BrainGate, so we have a collaboration with Jamie Henderson and Krishna Shenoy at Stanford, with the robotics group at DLR, and also Bob Kirsch and his group, who are the FES group, who are now doing BrainGate trials at Case Western. So with that, I'll thank everybody for your attention. So you want me to take some questions for a few minutes? So, sure. Yes, back there. You.
I was wondering, do amputees or people who lost limbs or became paralyzed at an early age also, do they have the same neural development or ability to control those missing or paralyzed limbs that the ALS patients would?
Yes, so it's not been done. It's not been looked at all, as far as I know. And it's an excellent question. That does the brain develop differently when it grows up without the usual apparatus? The only thing, there are two instances where I think give us some insight. People who are blind from birth due to fixable causes, and we'll take that as an example. And when they have their vision restored, they can't see. And they have to learn to see again. Things don't make sense. So the brain has to really restructure itself. And from what I understand from reports of individuals who have had this done is that they maybe don't see exactly like we do. They never get the same recovery. So we don't know for the motor system. If you grow up, it wouldn't surprise me. We just don't know.
Thank you.
In the back up there. I'm sorry. Yes. Oh, sorry. I'm sorry.
Hi, Dr. Donoghue. Under stimulation, you talked about memory loss, and what are you using there, and how does it affect Alzheimer's, and where's that going?
Yeah. So the idea of applying this was discovered accidentally. There was a trial going on in which they were stimulating the hypothalamus as a controlling appetite. And what they found, they did cognitive testing of this individual before and after, just to make sure they hadn't interfered with anything. And in fact, the person's cognitive abilities went up. So this has been tried in a number of rats and now in, I think, a half a dozen people. And there's an ongoing early stage clinical trial to say what happens if you stimulate. It's actually a bundle of fibers that connects to the hippocampus, a memory structure, and stimulating that to see if it can. So this is like DBS for Parkinson's. It does not cure the disease. It doesn't even stop it. What it does is it slows or masks its progression. So again, this is a trial. The idea is that most people develop the dementia in later years in life. If you could just stave it off for 5 or maybe 10 years, that the quality of life would continue to a point where other causes might might interfere with things. So, and for dementia, it's particularly important because it robs people of independence. They need huge amounts of care, so it's very expensive, and it's a pervasive problem. And that the number of people that will have some form of dementia in the next 20, 30 years is really a very large number. So this trial, as I said, is still early stage. It's not been fully evaluated. But there are early anecdotal, encouraging results, but we have to wait and see what happens. Now, where do I go next here? Over here, sorry.
This is a little ill-conceived question, so I apologize. But is there a way to compute either the number of electrodes or the size that would be not too big, but not, in other words, a bigger electrode would give you more information, a small electrode would give you more precise information? Is there a way to figure out what the maximum optimal size is?
That's an excellent question.
And one other addendum, if a lot of those muscle movements are facilitated with proprioception or sensory input, do you have a sense for what % of one of those patients' deficits would be enhanced if they had that proprioception?
So we don't know. It's a real puzzle. Some of our patients actually have proprioception intact and some don't, and we've not seen any difference. And we were surprised because if you have proprioception is your ability to close your eyes and know where your limbs are, and you know pretty well where they are in space when you close your eyes. And that information is not coming back, but in people that have it intact, not only is it not coming back permanently, it comes back, but it's not being changed when they try to move, which is what your brain would expect. So they seem to deal with that just fine. We think because your movement depends a lot on sensory feedback that it would be improved, but that there are people working on restoring sensory feedback as well. Oh, there's a mic back here, and then
Dr.
person here.
Dr. Donoghue, your appearance here today appears to coincide with a full-page article in today's Washington Post about an implant in a person that has a spinal paralysis, and this implant is supposed to, through all these mechanisms that you talk about, be able for him to control his fingers on one hand. The thing that surprises me so much is that the size of the implant is slightly larger than 1/8 of an inch square. I can't imagine how they could put that many probes in there in the brain in 1/8 of an inch.
So the 100 probes that we have are in 4 by 4 millimeters, so that's pretty tiny. I'm not sure I know the study you're talking about, but if it's the one I think of, they used a surface electrode, and there are a number of attempts acutely, just in short-term studies, to take signals from the surface of the brain and get just finger action. So it would just be one finger versus another finger. And that is sort of the kind of research that's going on to see what you can control from the brain. So I'm not sure I know that particular study.
It's
but
Ohio State and Battelle, I believe.
I don't know the study. Sorry. And over here, there was a gentleman with a question.
Thank you. You've worked with 9 subjects, and what my question is do we all run the same code, or is Kathy's code unique to her and couldn't be applied to, I forget, Matt? Would Kathy's code work for Matt?
No, we have to custom build the code for each person.
Right.
Because, first of all, we randomly sample. Remember, there are millions of neurons engaged in controlling your arm. And we sample a few dozen of those. And we randomly sample them. So we hit a different set in Kathy than we hit in Matt. And in us, those neurons have each individual neuron has its own sort of special properties, and it's special for each of us. So the hope of ever finding the similar neurons would be not possible.
So it's all about first learning the person's code and then applying.
That's right. So the first thing we have to do is do a calibration that's custom to that individual. There are changes which are probably part of the way the brain is normally operating that we have to cope with. That's one of the big research aspects that we're doing in signal decoding is what changes and what doesn't. We presume all day long as we learn things, as we're exposed to new information, that our brain is changing. And so we have to understand that. That's a wonderful neuroscience question, and it will also help build better brain-computer interfaces.
Thank you.
Oh.
Dr. Donoghue, this may be a little bit off topic, but I was wondering if we could get your opinion on some of these brain exercise programs that are out there, like I guess there's a Lumosity and things like that.
Yeah, so I'm not authority on, or do I know anything of any data on Lumosity? But what we do know from what was called the Nun study in Alzheimer's, which I know mainly through peripheral reading, but individuals who are have a more education level show less cognitive decline than individuals who have low educational levels. And the idea is that using your brain a lot somehow gives you reserve. And that fits with ideas that your brain is plastic and changing and you're always stimulating it, and that has a good positive outlook, and it also protects you against Alzheimer's disease. And that data is pretty solid. So then Lumosity is one of many ways that you could exercise your brain. As I told the students today, I thought maybe reading a really good book, a classic novel, or some mystery or something is something that would be equivalent to that, and perhaps to educators a more desirable thing to do than to play Lumosity. But so I'd say anything, from my own, not from a scientific perspective, but from my own just sort of general observations of what I know about the literature, anything you do to enrich your brain is going to help you expand the capabilities of the brain. But whether that particular system is, I said watch out, right? You just be careful of what you buy.
I'm curious about the codes, and the question is have you thought about tapping into somebody who has no damage, so that you can physically see what their hands are doing or such, and getting some ground data that way? And I'm also wondering, are the codes kind of temporal based? Is it that it's on, off, on, and then that means move the finger, or is it just you're tapping into which wire does what?
So the latter part, it's a little of both. It's so when you are moving, like handing that microphone, your brain is sending hundreds of thousands of impulses down a pathway that's a pattern of activation that engages your spinal cord, that engages your muscles in a very complex pattern of activation that gives rise to what looks like a very simple thing of just reaching over and handing somebody something. And the first part was the code and the...
have you tried it on somebody who hasn't had a...
The answer is no, because the able-bodied people are not enrolled in clinical trials like this. We haven't asked for permission because we wouldn't engage them. Whereas individuals like Kathy, early stage severe debilitation, and the FDA allowed us to engage those participants in a trial, but we wouldn't necessarily use able-bodied people in a trial yet. And there's no medical need for that.
Doctor, you've talked to us about sensors, and you've talked to us about the prosthetics that are being controlled. Can you tell us a little bit about the processing capabilities that are required?
So yes, a lot. The answer is a lot. It's sort of cutting edge of what can be done. Actually, the biggest boon to this has been the cell phone industry, which has produced very low power, high processing kinds of devices. So the little device that I showed you that Dr. Nermico had produced, the little implant, 5 years ago, that was probably not possible. The electronics were not far enough along. Now they are. So we're just at the edge of all the processing power. In fact, one of our rate limiting steps is the batteries. We have rechargeable batteries, and they won't last 25 years, for example, yet. They're not good enough.
Thank you, Dr. Wright. I'm encouraged, actually, that you have to do custom programming by subject, because it confirms that we are really individuals. But it degrades your research, but thanks. I was curious if Kathy's, I forget what you call them, codes or whatever it is, if they've changed over time. So maybe you did an initial translation and one bar meant left, and over time that may have changed, demonstrating plasticity of the brain.
Right. So there have been animal experiments that have suggested that's the case. But one of the things that this electrode array, we treat it as if it moves enough and the population changes enough that each day we just build a new decoder. It doesn't take long at all. So we haven't given anybody the opportunity to work with the same set of neurons every day. And that's something that we want to do because we think that learning will really make a difference in terms of getting better control, but we don't know how far that will go.
In order to advance the science that you're doing, what areas of technology would you like to see?
So the question is, since you didn't have a microphone, I'll just repeat it. What are areas where we need dramatic improvements? So it's basically in all of those areas. That's what I tried to list on that last slide or one of the later slides. So one is the sensor. Somebody said, would you want more electrodes? That was a question that came up. We probably, more should be better, but actually it's a neuroscience question. We don't know how many matter, but more, smaller, less invasive, more stable. We've seen some material degradation in electrodes that have been implanted for a long period of time, so better electrodes. The electronics, even though it's as small as a matchbox, we like it a lot smaller. The device, the cart of electronics that we want to shrink down to something the size of an iPhone, that's actually doable. It's just very expensive to do. So all of those things. And then, of course, a way to get these to people, which is it's very expensive because the surgery costs money, the technology costs money, to make sure that it's appropriate for people. And then, of course, the assistive robots and things are quite expensive. So we'd like to see things get cheaper, like cell phones have gotten cheaper.
Dr. Donoghue, recently a 114-year-old woman died, and they said part of the degradation was that she only had 2 stem cells left. What's the something that they measured her blood and that she was running out of stem cells or something? Do you like that thought? Do you like that
wouldn't
thought?
have any way to answer
that.
Sorry.
What is there any interplay or any interface on the stem cells, or are they at all related to the neurons in development?
So the encouraging thing here is for people with spinal cord injury, that there's a real interest and a long history of research to use stem cells to repair the damaged spinal cord. So if you could get it to grow, and it could actually reconnect the brain to the spinal cord. This is a really, really hard problem because you have to get the cut fibers to regrow, sometimes many, many inches, and then get them to reconnect in the right way, which may be very difficult to do. So those things are really not something that we expect to happen easily. They're at the point of being excited of axons growing a few millimeters across a gap in a human spinal cord. I think that achieving that would be a big deal.
I was going to let one of our students ask the last question.
Sure. Yeah, that's also a great question. So the barrier now is that the only way we know how to read out spikes, action potentials of the brain, which is the real language, the detailed language of the brain, is to put an electrode array into your brain. So I don't think there are many people who think that it's, that they would have that done just to have some additional control of something. And it's, as I said, I don't think we've reached the point where it's really so impressive you'd say, I need that extra control. So I don't think, in the near term, the idea of performing surgery to implant somebody with a device that doesn't add to their quality of life in a meaningful way or help them with a clinical condition is we're going to see anything. Now, on the other hand, there are those signals you can record from the outside. I showed a couple of devices. And there are consumer devices, which are little things you can put on your head, little caps, little electrode things. They record a very blurred vision of what's going on. And actually, many times, they record muscle activity because muscles also fire action potentials. But if you look online, there's one that's called Emotive, and people want to play games with them. And I've been surprised. They have been around for about 3 or 4 years, and they have not been successful. And partially it's because they're actually, you try them, they work kind of okay, and then people get fed up with them and they throw them away. So they're not really. But I would expect that games that can be played with signals outside the head would become popular. And just to give you a sense, the kind of signals you get outside the head are like being in the Goodyear blimp watching the Super Bowl. You can sort of get an idea who's on what side. You can get an idea that somebody scored a touchdown because you can hear a roar of the crowd. But you know, if you want to know what's the coach telling the quarterback, you have no idea. You'd barely know that they were even near each other. So it'll be a while, but there will be toys that I think. That's why I said watch out. They're fun, but they're not really doing what we're having to do.
So please join me in thanking you.
Thank you.